So our group was JZM, and our mutant of Brachypodium Distachyon was Bradi3g01830.1. So this is our original genomic sequence. It's very long, and using bioinformatics as well as reverse genetics and other methods within the Hazen lab, we were able to try to answer the questions, including what is the function of our gene and what kind of effects does the mutation have on our gene. So first we started by predicting the potential genes within our genome using F-Ganesh. And as you can see, it shows that we have 2 exons as well as 1 intron in between. And using phytosome, we were able to verify those predicted exons. As you can see, they're in pretty much the identical location. And at the bottom, the peaks where they are highest helps verify the location of those exons. Next, we found 5 different mutations within our gene, all on the 2nd exon. 4 of them are missense, as indicated by the yellow star, and 1 was our nonsense knockout mutation, as indicated by the red star, which is the mutation we focused on throughout the semester. And to the right of those, you can see the different amino acids that were changed with those mutations, as well as the change from nonpolar to polar or the other way around. Next, using PSI BLAST, we were able to find 7 different related species to ours, the Brachypodium Distachyon. And using these, we tried to find some articles based off of the function of our gene, but due to the lack of domains, we had to use these related species in order to find similar protein structures to the one that was present within our gene.
From there, we did DNA extraction and gel electrophoresis of our mutants to determine the concentration of our mutants through a nano drop, and as seen in our gel, mutant 5 and mutant 6 had the best expression of overall nucleic acid concentration. From this, we were able to do Sager sequencing of our 2 best mutants, which were mutant 5 and mutant 6, which were good quality. Both showed the location of the mutant as was originally found in our working map, that this mutation changes a C to a T. Mutant 5 is homozygous, as shown by the high peak in T. And mutant 6 is heterozygous, as shown by the fairly even peaks between the T, C, and A nucleotides. With these results, we were able to look more into our gene's expression overall and figured out that the gene expression is highest in roots and pinnacles. With this data, we were able to create a potential experiment using the data mining and the CVS data from the Hazen lab. With this, we determined a potential hypothesis that IAA would increase the gene expression in the roots of our plant compared to without any of those IAA hormone, and that would create a stronger gene expression of the root. We did this by planting 6 plants, 3 with control we sprayed with water, and 3 experimental we sprayed with 10 micromolars of IAA hormone, and then collected the root samples after 3 hours of treatment, making sure they were clean as possible, and then loaded the data into qPCR. From there, we did not get good enough results from the qPCR to determine the overall outcome of our experiment, but from the Hazen lab, we were able to determine with t-tests that the wild type and mutants had no significant difference on gene expression. With this, overall, we tried to understand the function of our unknown gene to help science know more about this mutation. This experiment was able to help us figure out a little more about many of our elements of our mutation. using bioinformatics and lab research results. Many of our elements of the mutation were able to be identified, but there were no domains that made it difficult to find the actual function of our gene. And through our experiments, lack of results also helped us figure out very inconclusive results.